Hey everybody, welcome to week 4. So this week we are going to be going through 3 different PowerPoint lectures. So I'm just going to bring them up right now. The first one is going to be based on molecular diagnostics. This one's going to be overall kind of a broad overview, but I'm going to make sure I highlight the things that I definitely do want you to take away from it. So we will do that here. Let me just get this into slideshow. All right. So within molecular testing for hematology, there are basically 3 main areas. One, to detect chromosomal translocations. That's essential for helping diagnose different types of leukemias and some other disorders. The second one would be just any other type of infectious diseases. So this kind of crosses over between hematology and microbiology. But these ones are specifically focused more hematology-wise: malaria, babesia, HIV. And then third is helping monitor disease after treatment. So different specimens we can take in for DNA or RNA isolation. That's kind of what it means by molecular testing. Molecular testing is all about the nucleic acid testing. So mainly DNA. We can do RNA, but DNA is usually more stable and easier to kind of isolate. So usually it's just DNA. So the following list at the bottom kind of gives you some ideas of samples you can take: blood, bone marrow biopsies, needle aspirates, and cheek swabs. So when we test the DNA, we're using enzymes and this whole replication process to make lots and lots of copies of that DNA sequence. Once we make the copies of the DNA sequence, then we have these probes that go in, and they're looking for specific malignancies, specific things that they've been targeted to, whatever we're testing for, essentially. At the bottom bullet, it talks about infectious diseases are detected by identifying, again, the presence of either RNA or DNA sequences specific to that bacteria or parasite organism, whatever it might be, like malaria is a parasite. So if we're looking for malaria and we think it's possible, there's a known DNA sequence linked with that malaria that it's looking for then. So this slide here is just background information. I'm not going to really test you on anything off this slide right here. So the biggest thing with molecular testing whenever DNA usage is being done is hugely critical to not have contamination. So we really must be very, what they say, meticulous, or very, detailed, very standardized testing, high focus, so we can really make sure there's no contamination, that it's a really quality test process. So polymerase chain reaction, or commonly known as PCR, is probably one of the main molecular testing methods. It is an enzyme-based method. Again, you're producing lots and lots of copies of that sequence, and so that way you can have a better chance of determining the sequence from that small amount of material. So what they do is they will denature the DNA. They will take it apart, because DNA is kind of 2 strands put together. They will take those strands apart. They read the strands. They're making the copies of each strand, and then they're looking for their set sequences. I'm just kind of doing a broad overview. Again, none of this is going to be on your test for this. This is just information on what it means to be PCR. So within the Pcr technique, there's always 3 controls required. There's a negative control, of course, a positive control, and then one that we call a no DNA control. That no DNA control's purpose is to show if there's cross-contamination. So if there's any sort of contamination that might show up, that would ding there. We would know. You have to run all 3 with every PCR run. So it's not one of those deals where you run controls and then 8 hours later you can run another control. No, they're run every PCR. So the different methods for detecting target DNA would be gel electrophoresis, the nucleic acid hybridization, DNA sequencing, and then probes for PCR. So this is just going to go through each one of these. Again, a lot of this is just background information, nothing too big that I really am going to test you on yet. So gel electrophoresis, hopefully if you've been through clinical chemistry, you've learned about electrophoresis. It's the movement of usually proteins right across an electric field, and then you can separate out those proteins to determine what you have. Kind of the same thing, you're going to have DNA moving across that electric field and being separated so that you can see the different parts of that DNA, what it's making. All right, nucleic acid hybridization, commonly known as southern blot. You might have heard of southern blot, maybe immunology or clinical chemistry at one point. There's a northern blot, a western blot, a southern blot. Southern blot is DNA-based. So again, you're using that jelly electrophoresis method that we just talked about to separate the fragments. And then it's going to go a little bit further and have a probe added, and it kind of has some extra steps to make it a little bit more than just the basic electrophoresis. So all you need to ever remember for our purposes as an MLT is southern blot is targeting DNA. You're using DNA in that method. And then DNA sequencing is basically within your DNA strand, there's a sequence of nucleotide bases, and you are looking for that set sequence, and you are going to kind of use that sequence in combination with the Pcr technique to make copies of them, to put in probes and determine if a set sequence is there. That's it. Like I said, very, very broad overview of molecular diagnostics. I always say that, within our degree, within an associate's degree of MLT, molecular diagnostics is becoming bigger and more prominent in the lab, but for our purposes, with everything that you have to get to know and learn, we just don't have a lot of time to focus on this. If you were to continue on and get your bachelor's degree after graduation, which I think would be amazing, then you would have probably more general science classes on molecular based. There'll be a lot of general sciences you would take to help beef up knowing about DNA, PCR, all that stuff. And then your core, like CLS classes, might contain more information on this. And when you work in the field, you might get the chance to be able to work with some molecular technology, and that is the best way to learn about it, is just being hands-on with it. So don't fret if this was a really quick overview. It's just background. You can read it a little bit further in your book, if you will. I guess to me, out of all of this, I think if I test on anything, it'd probably be that first slide there, the purpose of what is the purpose of molecular testing. So to me, that's probably the essential part. Hopefully when you work in the real world, you get to play with these instruments that are coming up and are being seen more and more frequently. All right, so we're done with that PowerPoint. We will go to, actually I'm not going to go to this one yet. We're going to go to the flow cytometry PowerPoint next. So flow cytometry is a specialized area in hematology. So not every lab will have flow cytometry. It's going to depend on the size of your lab. So if they're not a very large hospital, if it's not a large facility, say Minneapolis, it may or may not have flow cytometry. You might find flow cytometry more at reference labs or, again, a very large hospital-sized lab. But otherwise, like smaller kind of rural setting labs, you're not going to get flow cytometry. It's too specialized of a department, and it wouldn't be cost efficient to always have it, so they would send it out to another, like a reference lab. But basically, flow cytometry is a special analyzer that is going to look at, blood sample going through, and it's figuring out what cells are present to help diagnose malignancy. So that is the main purpose of flow cytometry, is figuring out what type of leukemia somebody has. So we can use bone marrow, blood, or tissue samples, again, lymphoid tissue samples, I should say. And when you put the sample through, that flow cytometer can detect 17 antigens on one cell simultaneously, which that's kind of freaking amazing. So it can look at all these different surface antigens, and each cell is unique with some of the antigens it will have. And by knowing what's on each cell, it determines what cell type there is present. So again, the purpose of this is mainly used for leukemias and a few other malignancy disorders. And so that will help diagnose it further. So the way that the instrument works is when you put the sample in, those cells are going to line up and go one by one in front of a light source. And as they go by that light source, they are going to kind of emit these fluorescent signals, and those signals are interpreted by the software inside of it. But the cells must pass individually to help be analyzed and detect the surface antigens. So as they go by that light source, scatter would also be detected, and scatter will help determine size and if the cell has granules. So forward scatter is going to tell us how big of a cell size it is. So it's proportional to the size of the cell. I do need you to remember that. That's important. Forward scatter will tell us size. side scatter is going to say the complexity, or if the cell has granules inside of it. That also is important to remember. So this is kind of a picture overview of what it looks like. So you see the little round blue things are each cell. So they're going to pass individually in front of that light source, in this case that laser. You have the forward scatter detector off to the left side. You have the side scatter kind of down to the bottom right. So those are going to, again, give a size and complexity of the cell. And then at the same time, there's these fluorescent signals that are being kind of bounced off and interpreted for the cell antigens and all the other parts of the cell. All that comes together to determine what cell type each one of these are as they go through. So once that happens, they will put together a dot plot. And they will represent how many cells of each type on this plotting. Now, you can probably find pictures of this in your textbook. That page number is not right. Maybe it's right. I don't think it is, though, so ignore the page number. But in this flow cytometry chapter area, there should be some examples of what a dot plot would look like and, you know, how they can plot it out in a picture format. But what we need to know is, okay, so now that we know that there are different surface markers on each cell and it helps determine what the cells are, we need to learn those surface markers. Now, there are a lot of surface markers. We're not going to learn them all. It just would be insane. But we're going to learn some key ones, and you do need to remember what they will be and what cell types they tell us. So first of all, we all have learned back in Heme 1 toward the very beginning, you know, we know that all cells come from a stem cell, and that we've also learned you cannot see a stem cell and like identify it under a microscope. Stem cells are what we call morphologically unrecognizable. So because we cannot identify them under a microscope, flow cytometry is absolutely necessary to help tell how many stem cells somebody has. So that's something they might do, like if somebody were to get a bone marrow failure, say like aplastic anemia, bone marrow failure, and they're suspecting that and they want to count, okay, let's see how many stem cells you have, and to see if that number is quite reduced or not, to know if that is a problem, is resulting back to their stem cells. They could use flow cytometry, look for the surface markers known for stem cells, and then figure out how many they have to know if somebody is low or not in that count. So CD markers, what we call CD markers, that's just the name of the antigens that are on the surface, so they will give them numbers. And so for stem cells, they are identified by the CD markers or antigens 34, 117, 38, and HLA-DR. So those are the antigens they know always exist on a stem cell surface. The one that I want you to remember is CD34. I do want you to remember CD34 is a marker for stem cells. So that's the first one that I want you to remember. So as these stem cells, you know, differentiate into other types of cells and form, they're going to lose some of these antigens, gain other antigens, and so these won't always stay. As they go to different types of cells and mature, we're going to gain other types of antigens on there and lose some. So, for example, blast cells, so like your myeloblast, pronormoblast, those ones, those really young cells, they typically have CD45 antigen on their surface. So I would remember that 45 kind of identifies some blast cells, some really immature blast cells. All right, so let's start with the myeloid lines. When I say myeloid lines, I'm meaning the neutrophil line, eosinophil line, basophil line, so basically your granulocytes. You can see the progression here from a myeloblast to promyelocyte to a myelocyte, and then there's some other steps in there we didn't put in. And you can see how they'll have certain markers, and then they'll lose some of them and gain others as they go. The ones that I want you to remember that would identify the granulocytes are CD13 and CD33. I always say think in threes. That's just the way I remember it. Yes, there is a lot more here that will identify a neutrophil or an eosinophil or a myeloblast, but because 13 and 33 are so prevalently found on that myeloblast, that's what I want you to remember. So remember, I feel like I'm saying remember a lot, but what we have to keep in mind is blast cells are very much look alike. So when you look at a smear, and you're seeing these large cells that look like a blast, you cannot tell me if it's a myeloblast or a monoblast or a pronormoblast. You would have no idea by just looking at that cell, because all blast cells look alike. They're all large. They all have that dark blue cytoplasm. They all have nucleoli. They all look alike at a blast stage. So because we cannot identify what they are, we just say they're blast cells. We'll say, oh, 90 blast cells seen, or 5 blasts. We can't really say specifically what type off of a smear. But if you were to send the sample through a flow cytometer, and it would come back and say, oh, there's CD13 and 33 on that blast cell, then we would know, oh, that's a myeloblast. That's when we can specifically say what type of cell it was. So for the granulocytes, for myeloblasts, remember 13 and 33 would identify like a neutrophil, eosinophil, basophil, those type of granulocyte cells. Monocytes. So again, kind of same thing. Monocytes share a lot of similarities with the granulocytes, and when you think back to your stem cell chart back out of chapter 7, the monocyte and granulocyte were under the same process for a long period of time before they finally split off, so they will share very similar markers. The ones that I do think that set them apart are the 14 and 15. So I do want you to remember 14 and 15 goes with the monocyte. Yes, they can share the 13, 33 like the granulocytes, but 14 and 15 will really help set that apart. So that's what I want you to remember for monocytes. All right, erythroid line. I don't really test you much on this, so we're not going to worry about it. Megakaryocytes, CD41 and 61 are really good markers there. I would remember that. So what I do is I say the bigger the cell, a megakaryocyte is our largest cell, the bigger the CD number. That's kind of how I keep it in mind. So 41 and 61 are the biggest numbers that we're going to talk about, and they go with the biggest cell. So I always keep that in mind. And then lymphocytes. Because there are 2 types of lymphocytes, we have both B and T lymphocytes, they are going to vary slightly. And we cannot, again, under a peripheral smear, tell if it's a B or T lymphocyte. We just know that it's a lymphocyte. So overall, both B and T lymphocytes have this marker that we call TDT. I need you to remember that. So TDT is a marker that's an antigen on the surface of both very immature B and T lymphocyte line. So it will tell you you definitely have a lymphoblast present, but it's not going to be able to tell you if it's a B or T yet. It's just saying, yes, there's a lymphoblast. There's an immature lymphocyte type of cell here. So always link that back to lymphs. Now, for the B markers are going to be 19, 20, and 10. Kind of said that weird, didn't I? 10, 19, 20. That's kind of what you need to know. So 19 and 20 tend to be more mature cell. I know the one says early marker, but 19 and 20 tend to go with more of a mature cell B marker. But basically just keep in mind 10, 19, 20 all will tell us a B lymphocyte. For T cells, I'm going to keep it super easy. It's anything less than 10. So if you look at it, 1, 2, 3, 4, 5, 7, 8, anything less than 10 is a T cell, is a T lymphocyte. It's as simple as that. So that's kind of in a flow cytometry. There is a lot to memorize here. One, you do need to know the overall process of how a flow cytometer works with that scatter, knowing the purpose of a flow cytometer, and then last, you have to know those CD markers that I just mentioned were necessary to memorize. So again, the purpose behind this is to help determine a leukemia, and leukemias are going to be affecting one cell line. Remember, when we name a leukemia, like acute lymphocytic leukemia, it's all about the lymphocytes with that leukemia. Or acute myelocytic leukemia, well, that could be maybe neutrophil line or monocyte line or red blood cell line. There's several in there, and we're going to learn the leukemias soon. In a couple weeks, we'll learn them. But this is getting to be starting on how we can help diagnose that leukemia. So last week we learned stains. This week we learned CD markers, and you're going to get case studies where it's going to give you stain results and CD markers, and you can help determine what type of leukemia it is based on those results, because it's going to tell you what cells are involved. So make sure you keep these memorized, know those CD markers. It's going to be really important, and it will help you out. And you definitely need to memorize them for test questions. All right, and then our final chapter PowerPoint here is going to be automated cell counting instrumentation. So basically it's just about hematology instrumentation. Now in the old edition they had this as an entirely separate chapter. I think in the new edition it's combined up with the coagulation instrumentation. So I think they put all instrumentation in one chapter. I could be wrong, but I know that in your book there's an instrumentation chapter of some sort and it will have this information in there. So let's go through what we need to know out of here. So there's two main principles to hematology instrumentation. There's electronic impedance and again scatter, kind of what we just covered with flow cytometry. We'll see that again. And you will need to know both basic principles. So first, electronic impedance, very first developed by Coulter. There used to be this, and there still is, this line of hematology analyzers called Beckman-Coulter, and they really used this methodology. And they were like the thing to have. So many labs had Beckman-Coulter. They were probably the most popular hematology instrument at the time. Now it's kind of switched away. There's newer, better technology out there, so you don't find them very much anymore. But it's still important to know how this electronic impedance methodology worked to count cells. Remember, it's all about determining the cell type and counting them as it goes through the analyzer. So what electronic impedance is they will have this small aperture, kind of like a hole, and on each side of that aperture, they will have electrodes. So there's one electrode on each side. As the sample goes through and the cells go through that hole, it will cause an impedance in that electricity of the electrodes. So there's like a current between those two electrodes, and as that cell goes through, it'll kind of break that current and cause this pulse to generate. And when that current pulses, they count it, and they'll be like, oh, that's a cell. There's another pulse. That's another cell. There's another one. That's another cell. And they count the number of pulses. So the number of pulses you have is the number of cells you had. So then the other part of that is how big of a pulse was it, the size of that pulse. That will tell us how big of a cell it was. And it can put together, okay, it kind of knows the average cell sizes of neutrophils, monocytes, lymphocytes, red cells, so it can use those pulse sizes to kind of know which cell it was that was going through there. And then that's how it gave you your results. It gave you the number of cells per each the count of cells per each cell type, and that's how it worked. So you do need to know electronic impedance there, and then all of that data, again, is plotted out. In this case, it's what we call a histogram plotting. And then what we just covered, which is flow cytometry, or what you can call optical scatter. Again, as the cells go by this light source one by one, it will cause a scatter of that light, and we measure that. Forward scatter, again, tells us the cell size. Side scatter tells us the complexity or granularity of the cell. All that data is put together so we can get the cell count on each type of cell, and again, it's all plotted onto what we call a scatter plot. That's it. Nice, short, and brief. Love it. So, all right. So you kind of have your gist of what you need to know for this week out of the 3 lecture PowerPoints. If you do have questions, as always, please feel free to reach out. Otherwise, have a fabulous week. Thanks.